Hey everybody, let's take a look at the nucleic acids, especially for those people that miss class on Tuesday. So nucleic acids, these are DNA, RNA, these are your information molecules. They're not only going to take information from generation to generation, but they're going to take information from the nucleus of your cell out to the working parts of your cell to produce the type of cell and the type of traits that you're going to have. Okay, very good. Here's a standard model of DNA, double helix. Okay, we're going to take a close look at its structures. How do these things work? This is referred to as the central dogma of biology. Okay, central meaning it's foundational. It is the crux of what's going on. Dogma is sometimes such a strict word. It's sometimes used in a religious sense. Catholic dogma. These are the rules that you just don't break. Okay? So the central dogma of biology, these are the foundational rules that just can't be broken. And that central dogma is that DNA stores material, RNA moves that message to produce proteins. Those proteins then are expressed as our traits. DNA stores it, RNA moves it, proteins express it, the traits are what we see. What's that individual's hair color? Okay. What's the muscularity on that horse? Okay. What color of coat does that dog have? The traits that we see outwardly in organisms. Here is an overview of the central dogma. So you have DNA replication from one for reproducing cells or from one generation to the next. Okay, so DNA stores it unless it's replicating it. That DNA then can be moved, that information can be moved via transcription to produce a piece of RNA that's an open code, okay, that can then be translated from the nucleic acid language into the language of amino acids. Okay, that translation then produces a protein. That is the workings of the central dogma. Okay. We'll see if we can't. Okay, so that central dogma then, let's take a look at where that goes from there. Okay, so DNA itself is a polymer of nucleotides. Nucleotides have three parts to them. Each nucleotide has a sugar, either ribose or deoxyribose, depending on if it's DNA or RNA, a nitrogenous base. Nitrogenous is just a big fancy way to say it's got a lot of nitrogen in it. Okay, and a phosphate group, typically a PO4 or PO3 with a charge. These are connected by dehydration synthesis to form what's referred to as a phosphodiester linkage. Okay, it's going to connect the phosphate of one group to the sugar of the next. So here you have the three parts of a nucleotide. Here's your phosphate group, your deoxyribose pentose sugar, and the nitrogenous base, in this case, guanine. Putting a series of those together, okay, here's 1, 2, 3, 1, 2, 3. Series of those nucleotides together will then form a strand, either one half of the DNA molecule or the entire RNA molecule. Okay. Notice we have the phosphodiester linkage, okay, the phosphate group linking together the two sugars. And we can also describe this in terms of the numbers of these carbon atoms. So we're going to number the carbon atoms starting at like 3 o'clock on a clock, just like we did for the carbohydrates. This is carbon atom number 1, 2, 3, 4. The fifth one's off the pentagon. Okay, we can describe those in such a way to say that things are moving or being produced from 3' to 5'. Okay, or we can describe the 5' side of that deoxyribose or the 3'